Got it.
જગ્યા બી એટલી જ છે.
Very soon, seminar will commence. Good morning, everyone.
Yes.
Respected Engineer Shri Bhuvai Patel sir, Chief Patron of this seminar, and President CDM University, Event Convener Dr. Bhakti Bajpayee, In Charge Head Arivas, Event Coordinator Dr. Ritu Dikshit, Organizing Secretary Dr. Nirmal Patel. Today's eminent speakers, Dr. Kishore Chikhalia, Associate Professor, South Gujarat University, Surat. A. Rawal, Professor, Chemistry Department, Sadapati University, Vallabh Vidyanagar. All the faculty members and dear participants. On the second day of this GUJCOST sponsored virtual seminar on pharmaceutical chemistry at the interface of 21st century and Indian perspective, I, Dr. Nisha Dakshini, on behalf of Haribas CBM University, would like to welcome you all. Yesterday, we had two very interesting and informative sessions by renowned speakers of their respective fields. Hope you all participants gain new insights in the field of pharmaceutical chemistry. Today's sessions are equally important as they are going to cover topics like importance of antiviral drugs in current pandemic situation and importance of heterocyclic scaffolds in therapy. Before we begin today's technical sessions, I'd like to give some instructions like each expert session is of 1 hour followed by interactive session of 15 minutes. And if you have a question during the presentation, we'll bring them up during the seminar and we'll also have time for questions at the end. Now, I would like to invite Dr. Ritu Dikshit, Head Pharmaceutical Chemistry Department, to welcome and introduce our first eminent speaker of today's seminar, Dr. Kishore Harkisundas Chikhalia, Associate Professor, with Narmad South Gujarat University, Surat. Dr. Ritu Dikshit, Madam, please.
Thank you, Dr. Nisha. Good morning, one and all present over here. On behalf of AntiHIV, I, Ritu Dikshit, welcome you all in a second day session of a 2 days national virtual seminar sponsored by GUJCOST, Gandhinagar. It's my privilege to give brief introduction of a Professor K.H. Chikhalia, sir. Presently, sir is working as a Professor of Organic Chemistry at Veer Narmad South Gujarat University, Surat. Sir is an active researcher and well-known scientist of a chemistry, and his area of research is a synthetic organic chemistry and medicinal chemistry. More than 25 students has pursued PhD under his guidance, and he has published more than 105 research papers in a national and international reputed journals. As per Google Scholar, his citation is 1275 and H index is 20. And apart from that, sir is also an active member of a various committee at university and state organizations. He has worked as an advisor for a UPSC New Delhi and also nominated as an expert in the field of higher education. Currently, he is appointed as Officer of Special Duties in a UGC section in Rusa State University level. Sir is an active member of a selection committees in a various university, BOS member, executive member in a various university too. We are very thankful to Professor K. Chikhalia, sir, for accepting our invitation in spite of his busy schedule. On behalf of AntiHIV, once again, I welcome you, sir, in this seminar. Thank you very much.
Good morning to one and all. Thank you very much, Dr. Dikshit, for a nice introduction. First of all, let me thanks to the AntiHIV Society for inviting me and giving me chance to share my thoughts with you. Earlier, I started some work on my area of research work is synthetic organic chemistry. Earlier, in my earlier phase of my career, I worked some on antiviral agents or medicinal chemistry. Perhaps right now also, I am doing some medicinal chemistry. But my area of research is a synthetic organic chemistry, organic synthesis through CH activation reactions. Everybody knows this point. It is a new frontiers in chemistry. It is a very hot cake, I am telling you, a hot cake in organic synthesis. Medicinal chemistry is my area of research. But what happens here, frankly telling, I am telling you that if you want to have publications, you have to starve a lot, because what happens, they required some, we don't have any in-house in vivo facilities. Some other crunches are there. That's why, or some other reasons, it's of interest also. I thought to develop some organic chemistry and I switch over to some organic chemistry. Still, I have been working on some building blocks of a synthesis. My papers are coming in a tetrahedron, Royal Societies, and like that papers. I'm not going to discuss these things, what I am doing something, but I'm a teacher and like to discuss with you regarding some antiviral drugs, because right, the current pandemic situation, some COVID-19 is there, virus is there, everybody's talking for a virus and people are going for a mask and something is social distancing is like that only. But before going to this disease, first of all, man has to go for a dissection of disease. That is very, very important. everybody's talking some malaria, viral, typhoid, something else said that. But before that, we should know what is the dissection of disease, what way we can identify the disease, we can dissect the disease, and on that basis, we can prepare medicines. Right now, in a current situation is what happens, if people are finding, see, I'm talking in general things. When I was a researchist, I was doing the same things only. Okay, if people find some beta-lactam structure, okay, that is a penicillin structure. So we are thinking, let me prepare some penicillin derivatives. Let me prepare some ciprofloxacin derivatives. Let me suppose 3-member ring is there in organic structure. So what we think, let me prepare some, were thinking to enter a 3-member ring. Those are just trained rings without seeing anything. People are thinking that some nicotinic acid derivatives are there, or anti-GUJCOST drugs are there. We start working with the INH, isonicotinic acid hydroside, and we just concentrate our research on that direction. But friends, I'm telling you, yesterday I had discussion with Madam Dixit. She told me that some young faculties and research students are there. So I thought it is very interesting to know. It is interesting to interact, and we can think it. What way I can disperse, or I can share my thoughts with you. And that's why I'm discussing with my thoughts. Let me start some presentations. I'll start some of my presentations. So I'm presenting my work in a prior PowerPoint. Just 1 minute. Yes. Yes, please. So my topic of my talk will be life cycle of virus and related antiviral agents. Actually, what happens here? First of all, what in the first way I told that it is a antiviral, anti-HIV chemistry. We should know the dissection of disease. What way we can dissect the disease? So I'm telling you just a glimpse of this virus, anti-HIV, and from this anti-HIV, we will discuss what is antiviral coronavirus. That's some of the slides I have prepared, and what is remedies. Something like that I have just shown this thing. I'm thinking to show this chart with you. Actually, what happens? Virus is there. I use this one word. Okay, virus is a very intelligent. It's a brain without body. Brain without body. In our computer also, you find virus is there. So you can say we are getting so scared or something disturbed, something problem is there. We are going for antivirus devices and like that. So virus is I use this word, it's a very intelligent, because viruses infect the body, immediately it gets replicated. And as it gets replicated, a new virus is generated. So again, new virus is generated, again it gets replicated, it is going to other strains, and keep on going on, changing its original structure. And as we don't know the structure of virus, we cannot prepare the medicine. Because any medicine is to be prepared, you know that targets we know, okay, this is the disease only for which I prepare the drug. But we don't have any target, we don't have any idea what are the structure of virus. And unless we know the structure of virus, we cannot prepare any medicine. Because see, one structure is identified, and if we want to prepare, if we want to prepare these molecules, we design the molecule. If you go for some simulation study, if you go some molecular modeling study, and you just think it, okay, this terrorist compound I will prepare, and that will have a probable antiviral activities. But it is not happen. Once structure is identified, virus is what its nature, it gets replicated, and new virus is generated. And again, it is going to one stage, again gets replicated. So people do not know where it stands. So we don't get the structure of a virus. And that's why it is very difficult to prepare antiviral drugs. You might have seen SARS, numbers of viruses, I don't say their name, but numbers of viruses came in this earth and they went away. Their autocoids are generated in the body, and that helps only in combating the virus diseases. These are the things I'm telling you. And other things, if you know the identity, if you don't know the structure, then we find out, okay, what is the root or who is supporting the virus. So one group has identified, or researchers has identified, enzymes are there. Enzymes are supporting virus to go from one group to other, because enzymes are a catalyst of our body. And you know that we are taking the foods and everything, we are getting metabolized. They are going through various processes. That is why who is catalytic? Catalysis enzymes. It activates energies, increases. So it is this activation energy concept everybody is knowing. So this catalytic activity of enzymes that helps in digestions and everything. So in HIV also, I have just gone through these things, and I will just prepare slides for this one. And we have prepared some of the compounds that I have not. I may show you in this slide also.
Interrupt you, sir.
Yes, please.
Sir, screen is not visible. Is it presented or still?
Screen is not visible right now. Not visible.
No, sir. No, sir.
Oh, 1 minute. Please. 1 minute. 1 minute. Now it is visible?
No, sir. Still it is not visible.
Still not visible.
Yes, sir. Yes, sir.
Just 1 minute.
Yes, sir. Yes, sir. It is visible now.
Okay. That's all. Is it okay now? Now you are able to see?
Yes, sir.
Okay. That's okay. So virus is that's why I use this word one. You know the example of this cuckoo and crow. A cuckoo puts her eggs in the her nest. Crow hatches it, and what crow is thinking, my generation is coming, but the generation is of cuckoo only. Likewise here, virus is infected the body will not know anything, so what is happening, and a new virus budding is there, and then you come to know, okay, somehow I am infected with things. So likewise, I'm sharing thoughts with her anti-HIV chemistry, that is HIV life cycle. First of all, you know that should know the dissection of disease. So in order to virus, I will be targeting the enzymes. What way we can inhibit the growth of enzyme, that is very, very important. So I will simply read this point here. In order to see, I'm just telling first thing, HIV virus is infected to our human only. Why? Because HIV has got more affinity towards the CD4 cells. The CD4 cells, that is called, is a T4 cells. CD4 cells, CD4 receptors. The CD is more responsible is what? T4 cell. The T4 cell is responsible for its warming of your body, immunity, warming of your immune system. But that is the best thing only. So in order to virus to reproduce, they must infect a cell, and a virus is also technically alive. See, people are telling it's alive. Virus is not technically alive. It is a protein capsule only. So they're sort of like a brain with no body. In order to make new viruses, they must hijack a cell and use it to make new virus, just as our body is constantly making new skin cells or new blood cells. Each cell then often makes new proteins in order to live or to reproduce itself only. You know that body has already constantly preparing salt. Various new cells are generated. It's a constant process. Now, virus, when body is infected with the virus, virus hides its DNA into DNA of the cell. This is a very important point. The virus hides their own DNA into the cell, into the DNA of a cell, and then when cell tries to make new proteins, it accidentally makes new virus as well. Because body is constantly preparing new cells, virus is hiding its DNA, which is matches to our DNA, and body is constantly preparing cell. Body do not know I am infected or not infected, but they are producing the viral DNA is generated, and that is HIV mostly infects cells in immune systems. And infections, several. See, I'm talking for SARS. Also, one virus has come. Suppose HIV is there. I'm telling you in a very our language. The virus is there. It is affecting to animal. Animal is not affecting because animal has no CD4 cell, no receptor, CD4 receptor. T4 cell. This is a CD4 receptor is on human being only, and that's why it's infected to the human only. animal is not infected because they don't have CD4 receptors. And that's why infection, several different types of kinds of cells have proteins in their own surface that are called CD4 receptors. And HIV has more affinity towards CD4 cells receptors, and that is people is affecting with a T4 cell is responsible for infections or meeting with a HIV. And virus is what? It is like once HIV replications. What I told you, virus infected the body, it gets replicated and new virus generated. So you don't know the structure of a virus. And that's why likewise, once HIV binds to cell, it hides HIV DNA inside the cell's DNA. Then this turns, one hiding is there, body has its own DNA. So this process keep on going on, and that's why becomes our whole body becomes a virus factory. HIV factory, it becomes virus factory. Now, to understand HIV, we have to go for several definitions. I have just prepared some definitions. Just listen this point. This is I have prepared on my language only, my level or my language. What way I can understand HIV infections. The DNA is there. DNA is the main portions because it is being hidden and new DNA is generated. The DNA is like a blueprint for building living cells. Enzymes are the workers of a cell. They build new proteins, transport materials around the cell, and carry out other important cellular functions. RNA is like construction boss. Cells use RNA to tell enzymes how to build a specific part of cell. To make new proteins, enzymes will occupy a specific part of the DNA into pieces of RNA. This RNA is then used by other enzymes to build new protein or enzymes, because cycle is just like going on. Proteins, we know the proteins things. Proteins, building blocks, those are used to make living things. Nucleus, a small package inside the cell where the genetic material is kept. So these are the 5-6 definitions I told: DNA, enzymes, RNA, proteins, and nucleus. So entire study for entire virus structures, virus study, we have to remember these points only. That these are the main portions or main things to understand life cycle of a virus. So this is the first thing I'm telling you. What I told you, the first virus is infected the body, our human being, because human is a CD4 receptors, which is having a T4 cells. It's got more affinity towards the CD4 cells. The CD4 cells is in humans only. So this is first is binding. What way is a binding? What type of things are there in the binding? That I'm not going to discuss this point, but it's a binding. First infected the body. The virus consists of other outer oral envelope of protein. Virus itself is a protein. If fat and sugar wrapped around this set of genes, in case of HIV, genetic information is carried out in a RNA instead of DNA and special enzymes. HIV has a proteins on its envelope that are strongly attracted towards the CD4 receptor on the outside of the T4 cell. While HIV binds to the CD4 receptor, surface receptor, it activates other proteins on the cell surface, allowing HIV envelope to fuse to the outside of the cells. I'm just telling this point. Just like in army, the invaders is coming. The first single signals are there. On the frontiers are there. They're just combating it. They are opposing it. Like on our body also, we have a CD4 cells. But other things, when HIV is just coming to the body, our other cells are there opposing it. But when it meets the CD4 cell, its attraction becomes very fast and it try to interact. And when they interact with CD4 cell, immediately it gets fused in the body. And once it's entered, then it starts its work. It hides its DNA, and that DNA is hiding its DNA, and DNA is getting constantly preparing our cells, and automatically body becomes a virus factory type things. So that's why I'm telling you more emphasis given to CD4 cell. Now CD4 cell is called it is entering. Then who is supporting? First, in the first slide, I told you that it is a brain without body. It is a protein capsid, is not living, but enzymes are there. Enzymes are supporting. So first I'm telling you, friends, enzymes, we don't have to kill virus. It is a non-living, but we don't have to concentrate on virus only, but we have to find out the enzymes. identify the enzymes, which enzyme is supporting the virus. So we don't have to do kill virus, but we have to inhibit the growth of enzyme. And if enzyme is inhibited, our works get over. The virus will not replicate because it's a supporting or catalyst. We are blocking the catalyst. That's why that's the things only. So that's why I'm in my entire talk, I will talk about the activity of enzymes only. Okay, what way we can we can inhibit the growth of enzymes, or we can block on the enzymes. That point I'm just telling you in entire my talk. That's why my focus on drugs will be based on a enzyme structure only. So once a virus is entered, so this all various stages are there. I will be talking about 4-5 stages that you must be knowing, because all pharmaceutical people must be knowing: reverse transcriptase, translation, protease, integrase. They are all enzymes. So I study on enzymes only. Some of the enzymes I have identified, some of the stages I have identified, and that I'm going to talk with you. Okay, first enter virus is entered. An entry can be blocked by entry inhibitors. Various structures or various components, various drugs has been identified which can stop inhibiting or entering the body. The entry inhibitors. The entry inhibitors work like by preventing HIV from entering the healthy T cells in the body. They work differently than many of the approved HIV drugs. See, I'm telling you, all drugs which is prepared from virus, I said this is serendipity with synthesis. Just like nevirapine was there. Nevirapine was a drug. It was a drug that was prepared for anti-cancer. And then it has not worked so beautifully with anti-cancer, but it has worked for HIV. Likewise, I'm telling you, it is serendipity with synthesis. So they work differently from this many of the approved HIV drugs. The protease inhibitors, nucleoside reverse transcriptase inhibitors. One example I'm telling you, nucleoside. Our body must be knowing nucleoside, nucleotide. One nucleoside is there, nucleoside transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors, which are active with infected cells. Yes. Now, second point is the reverse transcriptions. HIV genes are carried out 2 strands of RNA, while the genetic material in human cell is found in DNA. In order to the virus to infect the cell, process called reverse transcription makes a DNA copy of the virus RNA. You understand spike, DNA copy means copied from virus RNA. After the binding process, once it's binded, the viral capsid is a protein only and is released in this host cell. When bind viral capsid, it's released in the host cell, and a viral enzyme called reverse transcriptase makes a DNA copy of the RNA. Now that new DNA is called proviral DNA. You know that point where there's a side its DNA. The body does not know anything, but which viral DNA, which direction? Hello? After the binding process, the viral capsid, a viral enzyme called reverse transcriptase makes a DNA copy of the RNA. This new DNA is called proviral DNA. Reverse transcriptions, this is proviral DNA is generated. Now, one enzyme, this one is a reverse transcription, can be blocked. This process can be blocked by nucleoside reverse transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors. Next is a nucleoside and nucleotide. Everybody knows this one, nucleoside, nucleotide. You know, this is a simple nucleic acid, nucleotides, nucleosides, we understand is quite 3 days, 2, 3, 5 phosphates, we understand is quite very well. The nucleoside analogs act as chain terminators or inhibitors of the substrate binding site of RT. NRTIs, that is nucleoside reverse transcriptase inhibitors, must be phosphorylated. This is simple phosphate groups, everybody knows 2, 3, and 5 positions. 3 steps to their 5' triphosphates to become active inhibitors. Nucleotide analogs, that is nucleotide reverse transcriptase inhibitors, already contains a phosphate group. There is 2 steps only. Earlier was there, nucleotide is there, it's over 3 phosphates, 3 steps only, because it is already phosphate groups are present. This 5' triphosphate of the NRTI competes with 2-deoxynucleosides, 5' triphosphates for binding to the reverse transcriptase, leading to the viral DNA chain termination. Okay, some of the nucleoside drugs are there. You must be knowing acyclovir, ganciclovir, nucleoside drugs. These are the drugs working. See, you must be knowing this point. Acyclovir, ganciclovir, we can go for it's a pro-drug concept also. There is a different things only. So these are the currently 7 FDA drugs and nucleoside. This transcriptase are this one. Judovidin is the first drug. It was first discovered. Then diaminosines, gictovidins, stuvidins, luvinidins, abaclivir, and right now tenofovir or vir-containing compounds are there. There you can say all are nucleoside analogs. Non-nucleoside. Non-nucleoside has a no function, we can say. No function, no. I'm not telling this no function, but it is like any structures. Suppose people are going for a efavirenz. See, this nevirapine contains 3-membered ring. Efavirenz contains 3-membered ring. So these are the two hot favorite drugs, you can say. So we are the initiators of the researches we find. A 3-membered ring is responsible. We understand the chemistry also. A 3-membered ring is highly strained. You must have studied Baer strain theory. 3-membered ring highly strained. So what is our perception? If 3-membered ring, the compound is going to the body, it can change some allosteric site. It can go for allosteric site, can be changed. It can change the conformation of enzymes, and that's why the growth of enzyme can be inhibited. Like this is our perceptions. Friends, I'm telling you, you see the structure of a anti-HIV drugs. Various peoples has shown their views. Some people in the earlier phase of 2003 to 3 or 2 to 4, 2002 to 2004. You must have seen Ukum was there. And Ukum was publishing the papers, numbers of publications on anti-HIV. I used to say he was working constantly for anti-HIV, particularly for a non-nucleoside reverse transcriptase inhibitors. Once in one structure, one paper he has shown DAPI, DAPY, diaryl pyrimidine structure. Other structure he has shown, one group has shown it is a PETT analog, phenyl ethyl thiazolyl thiourea. One group has shown it is a horseshoe type. You know the horseshoe type structure that is also responsible for anti-HIV activity. One group has showed it should have a heat pH NHU, heterocyclic NH, phenyl and unsaturation. One group has shown it is a PTT analog. One group has shown some ideas of a butterfly structure. So likewise, various peoples are taking on a different concepts. Suppose my students is showing me, yes, sir, cyclopropane ring is there. So I always tell them, yes, cyclopropane ring has a good activity because of this, is a best strain theory. It strains, because of the strains, it inhibits, it distracts the structure of enzymes, so it will be better. Then my contemporary will say, no, it is not like that. Just like prepare a butterfly type structure. So we prepare the structure compound showing the butterfly structure and we try to synthesize whether it inhibits the growth. But every people is going for trial and error, but nobody is getting success because we don't know the structure of the virus. That's the problem only. So it's NRT do not require bioactivation kinases. And NRTs are currently, this is the 3 drugs are there, but basic structure is this one. Cyclopropane, it is a very hot favorite right now. Now this is based on this one. Several drugs, these drugs are in the clinical trials. Tivo, Lovir. This I am talking for. This is the name of the drugs which I am telling. That is of a simple clinical trial. These are going in phase 1, phase 2 clinical trials. Some drugs are approved also, but not surety for any virus. I am telling you general things. I am telling you. Next point is of integrations. So the integrations, the mathematics we are going for differentiations and integrations like that only. The HIV DNA is then carried out to the cell's nucleus, where the cell's DNA is kept. Then another viral enzyme called integrase, one enzyme. As means it is all enzymes, which hides the proviral DNA into the cell's DNA. Then when the cell tries to make the new proteins, it accidentally, when body is constantly preparing the cells, so it can accidentally make a new HIVs. And integrations can be blocked by integrase inhibitors. Every class has a different drugs. It is a trial and error only. Drugs are prepared. Then next is a transcriptions. Once HIV's genetic material is inside the cell's nucleus, it directs the cell to produce new HIV. The strands of the viral DNA in the nucleus separate these special enzymes, create a complementary strand of genetic material called messenger RNA, which instructs the HIV to prepare a new HIV. Then transcriptions can be blocked. Again, this is one enzyme. Transcription is one of the enzyme. The transcriptions can be blocked by antisense antivirals or transcription inhibitors, the new class of drugs that are in the early stage of research. One is the translations. Translation, everybody knows what is translation. It is as is binded, reverse transcriptase, integrase, then generation of a messenger RNA. The messenger RNA carries instructions for making new viral proteins from the nucleus to kind of workshop in the cells. And each section of mRNA corresponds to the protein building block for making a new virus part of HIV. And as each RNA strain is processed, a corresponding string of protein is made. The process continues until the mRNA strands has been transferred or translated into new viral proteins needed to become new viruses. So these are the, by summation of the stages, there is a virus assembly and maturation. See, every stage, once virus is entered, then met with a nucleoside transcriptase, then integrase, translated, mRNA generated, then whole things. It is a budding, I say it's a budding or virus assembly and maturations. The minor stage becomes with the assembly of new virus. Long strings of proteins are cut off by the viral enzyme called a protease into the smaller proteins. Now these proteins are serve a variety of functions. Some become structural elements of new HIV, while other becomes enzymes such as a reverse transcriptase. Once the viral particles are assembled, they board up the whole cells. The whole body becomes a virus, and it creates a new virus. New virus is there, again gets in, enter, same cycle is going on. So our whole immune system gets collapses. And then body comes to know that our immune system get collapses. Then a person happens to go to medicals. And then they are telling you are infected with a doctor. Medical will not tell you immediately at one moment you are infected with the HIV, but they will ask to go for treatment of a tuberculosis, because somewhat extent tuberculosis and viruses properties are a little bit resembles to each other. And then it is confirmed by tuberculosis, then the doctor will tell you, okay, you are infected with HIV, like that only. The virus enters the maturation stage, which involves the processing of viral proteins. Maturation is the final steps in the process and is required for the virus to become infectious. Viral assembly can be blocked. Whole assembly, once assembly is there, the whole system is gone. Then again, also some of the remedies are there, some medicines are given, which is protease inhibitors. That is, maturation is a new target of the companies developing a new HIV drugs and they blocking these protease inhibitors. Now, protease inhibitors, against this is the thing. So during the production of a cycle of HIV, specific protease is needed to process a gap pol. You must be knowing these proteins, polyproteins, into mature HIV components. If protease is missing, non-infectious HIV is produced. And then people have started, see, our chemist, what is the function of a chemist? See, virus is just like doing this business, going to one stage to one strain to another. That's why some of the researchers, active researchers, they found, let us develop crystal structures of enzymes. And enzyme structures is prepared. So then we can find out exactly what is the positions, what portions, where we prepare the medicines which can block the activity of enzymes. The crystal structure of HIV protease was first obtained by Merck Laboratories. Now this is a structure of this one, and this is protease is 99 amino acid aspartyl protease that function as a homodimer with a one active site. See, these are the protease inhibitors. These are drugs. I'm not going to tell the drugs, but what chemistry is there? That is that I'm telling you. One is again what I told you. Many drugs are synthesized. That is of a serendipity synthesis. One of the compound is a quasoline. Quasoline was originally developed as an anti-cancer agent by researchers at Purdue University at National Cancer Institute. The quasoline was developed from a chemical known ATA, that is anti-renotearcarboxylic acid, which has long been known as anti-HIV activity. These are the quasoline is there, but now, yes, right now, people are going for HAART. One word is very common, that is HAART, HAART, highly active antiretroviral therapy, is a standard care of people for HIV. One more things I'd like to do, we have to go for a toxic, because time is going off, I cannot complete these things, that's why I'm just keeping this point only. The new toxicity and the side effects, all antiviral drugs are toxic. Toxicity point is to be measured. And all drugs has one property, they are going for admit this one, absorption, distribution, metabolism, excitation, toxicity, reducing drug toxicity, the use of combination therapy, the combining agents which are favorable synergistic properties allows a decrease in dose during dosing frequency. Ritonavir alone cause gastrointestinal side effects, but when used in combination with other PI, it can be administered at lower dose. Okay. Now I'm just focusing this point on a NNRTIs, non-nucleoside Use transcriptase inhibitors. I'm just skipping this point, but I'm just telling this one point. Yes. See, this is a very interesting point, friends. What I told you, you don't identify the virus, but you can identify one enzymes, which enzyme is responsible for this, for replication of the virus. So that's why we don't have to go for concentrating the virus, but we have to go for concentrating the enzymes. So one group, you have prepared one crystal structure of enzymes. The crystal structure of enzymes, I'm just repeating this point. This is a crystal structure of enzymes. You can just like a hand, one hand is there, right hand, where you can find fingers. This is a, yes, this is a fingers, primer, template, thumb. This is RNase H, like this NRTS. So we just prepare these things. You can see in our hand is a, all I shown you this one, purple color is a primer, template, fingers is there, where I can design a molecule. I can design a molecule, and I study this point. If I design the molecule, it can be exactly binded to certain sites. So virus cannot be cannot be supported. It cannot go out. It is just getting, its growth can be inhibited at this point. This is a structure development is very, very important. And based on this structure, various group has, now I'm showing this point here. Yes, these peoples has prepared, it is found, okay, these medicines are possible. I'm just reading this point. Yeah, don't read whole points, but NNRTIs bind to NNRTI binding pocket, where allosteric hydrophobic site close to what distinguishes and the catalytic active site of the RT. As they are inhibitory against RT transcript as such, NNRTIs do not require intracellular metabolic activation, but it is a non-competitive inhibitors with respect to the subtraction, non-competitive or uncompetitive inhibitors with respect to the template of primers. This is all drugs which is prepared that is based on a, this is what crystal structures. See, I'm telling this point also. One structure is DAPI is there, this word, DAPY, DAPI. This is diaryl pyrimidine. See, very interesting point is what every group who is working for anti-HIV, they are just making a trial and error. One group has shown isopropyl group, other people say isobutyl group, likewise this one. One group has shown that the structure should have a diaryl pyrimidine, in this case a butterfly structure, so people start working on the butterfly structure. But activity is different. In our India, you must be knowing this point. One person is working for nicotinic acid, other immediately starts with isonicotinic acid, other is starting from other things only. But it's not like that. We are just making trial and error. But trial and error, it should be of a logical things only. So this is the personal, see that time we have to go for a chemistry. The personal flexibility of the inhibitors can generate numerous conformational variants of the compactness of the inhibitors permits significant reposition of the and reorientation of a drug. So I'm not reading this point, but just I'm telling one simple thing. What type of the molecules are required? Yes, that I'm telling you. These are the very, all drugs are prepared. See, one people has shown efavirenz, delaviridine structures. That is of a cyclopropane. Other analogs are same, but only the change is, see, other analogs are differing, but only one analog that keeping a cyclopropane ring is there. One group has shown it is a phenyl ethyl thiazolylthiourea, PTT analog. One group says HEPT is there. One is a quinoxylin derivative is there. Various groups are showing various things only. So in this case, we are going for a competition study also. We are finding the non-inhibitory binding pocket is there, where enzymes are binded. What is the score? That all preliminary things or preliminary parameters we have been studying, but that is not so justifiable. I'm telling you, and that's why doctors are advising to go for a heart therapy that is of a highly active antiretroviral therapy. Now, one group has shown dial-triazine-based derivatives. That's data, diaryl-triazines. So these structures, data derivatives, means diaryl triazine derivatives. This is one compound data, diaryl triazine. You see this molecule you are taking triazines. One side you are entering aromatic group, other side you are entering aromatic group, and you see this derivative is there. It may have activities. One analog is triazinyl amine analogs. Thiourea is there. PTT analogs are there. Cyclic compounds are there. Cyclic thioureas are there. Isothioureas are there. Numbers of things are there. When people start preparing the derivatives, perhaps I also work on this compound. I have prepared analog of these compounds, and we tested. I did collaborations with a Riga Institute Belgium, where my laboratory, my students used to synthesize, and they were sending it to that Riga Institute, and they got some activity. Some results we got it. Some promising results also I got. Like in my group, we got it. But problem of toxicity, because we are our culture, our students in our laboratory conditions, they won't tolerate. They want to go for. We don't concentrate more for toxicity, and because of the very good inhibitions activity, we got it. But toxicity has disturbed it, so selectivity index becomes lesser. So it is not a promising. We cannot say it's a promising drug. Perhaps we designed in such a way, perfect way, we got the inhibitory activity, but because of the toxicity, we couldn't reach up to the goal. But at least design the molecule in that directions, and we got some promising results. These are the from compounds. Like PTT, I'm not going to discuss this point, but important find. See, your pharmaceutics peoples, we can see if one molecule, this is one derivative, is a drug of choice, this compound. What way we can go? If we can go for a disconnection. Is a chemistry is coming by disconnection, we get one analog. This is again disconnections, we get this analog. Again disconnection, we can get this compound. And this is our final preliminary compound, we got it, and then we can go for synthesis of these molecules. So this is a simple things. What basic structures I've shown you here. If PTT analogs are there, if they have got activities, I can share, and you can take my PPT, but study it, what is written actually exactly, and what we intend to say, what is our message, that we can find it. But let us discuss some other points also. Damni derivatives, so just I'm just highlighting the things only. One damni derivative is there, diaryl methane type analogs. You must be knowing this is a metronidazole drugs, metronitromidazole derivatives. That is called damni derivatives. On the basis of molecular modeling and extra structure, investigations of the neirepine and TIBO, Skefferedal proposes a three-dimensional model describing the structural elements that were dominant for anti-HIV activities. The structure I'm showing here, yes, this is the things only. This is we can shape these molecules and these reactions. When nevirapine is there, TIBO is there, these drugs are there. What is summation of all these drugs? What is the basic tentative structures we find? The basic structure is this one. So summation of this one, friends, we come to this one Davos, the derivatives, this compound. We can go for disconnection. We find what is the structural analogs. This is analogs. This is also improved damnis. Again, this is metronidazole derivatives are the structure. And this is the main framework. So what are significant points we find? What could be the activities, why the activities increase, or what is the reason for getting the hydrophilic region, lipophilic region, where we introduce this lipophilicity. So we can say one is a benzene ring, one is extended pi systems, is associated with the thiocarbonyl groups or thio groups or carbonyl compounds, mercaptan compounds, or you can say methyl groups. And these summations can lead to a lipophilic regions. That lipophilic region, we just designed, this is a metronidazole, metronidazole derivatives, and that has some fragments like this, and which leads to the our direct target, that it should have a butterfly structure, it should have a PTT analog, it should have a horse type structures. These are structural possibilities we find, and we can synthesize these molecules. Now, this is the simple thing. Likewise, it is of a COVID also, the function is the same, coronavirus. This is a virus. Virus we understand. Every people, they are talking for what is COVID. See, whenever we happen to go in some places, people are giving a mask and that COVID is there, COVID is there. Actually, what happens? I'm telling you, friends, actually, coronavirus 1, it was discovered in 2013. You see the research papers, 2013 was there. But right now, why is it so pandemic? Because it has got one enzymes that is of a ACE. Everybody knows this one, angiotensin converting enzyme, ACE. ACE has got binded, this substance is found, but it has gone through pulmonary attack, this going for a pneumonic disorder, that leads to our COVID. Actually, this is the only things. So I have just prepared some slides I'm showing here. Here in this case, COVID-19 pandemic is also known as coronavirus pandemic. Everybody knows this one. It is a preliminary things. I'm not going to read it. Actually, on February 11, the World Health Organization named this disease a coronavirus disease 2019, COVID-19, which is similar to severe, see this word, severe Middle East respiratory system, that is MERS syndrome, and severe acute respiratory syndrome in protein sequences. In early December, a novel coronavirus detected. This I'm not going to read it. Okay. Actually, what is coronavirus structure? Everybody, you are pharmacy people, everybody knows this point. The coronavirus belongs to the coronavirus family in the nidoviral order. Corona represents a crown-like spikes on this outer surface of the virus, which was named as a coronavirus. Actually, this is this nucleocapsid protein, envelope glycoprotein, RNA spike proteins, membrane. This everybody knows structure. I'm not going to read the structure. But important is what this is for. Yes, I want to distinguish this point. The subgroups of the coronavirus family are alpha, beta, gamma, and delta coronaviruses. The severe acute respiratory syndrome, H5N1 influenza, H1N90 Middle East virus, this acute respiratory distress syndrome, which leads to the pulmonary failure and result in a fatality. These viruses were thought to infect only animals. Keep this word in mind. These viruses were thought to infect only animals until the world witnessed the severe acute respiratory syndrome outbreaks caused SARS-CoV 2002. See this point in Guangdong, China. This one. Only a decade later, other pathogenic virus known as a Middle East respiratory system caused endemic in Middle East countries. See the story. This is the talk of it 2002. Now this was me knowing alpha coronavirus. One is of a beta coronavirus. One is a gamma, and one is this is a delta, and one is a delta, and this is a gamma coronavirus. This also everybody knows these things. Now in this case, important point which I'm not going to read these all things, but you must be knowing this S protein and RBT ribitide binding the domains region of SARS-CoV-2 has enhanced transmission ability. So this point is very important right now. Okay, global research efforts have already revealed that novel coronavirus is an overlapped envelope virus. As the same name implies, virus envelope have an outer covering shape envelopes their genetic material. SARS-CoV-2 virus enters the human. See this point. SARS-CoV-2 virus enters the human through the respiratory tract and starts its actions after binding the angiotensin converting. This is very, very important. ACE2, angiotensin converting enzyme. The virus has 4 main structural points: spikes, protein, membrane, and nucleocapsid. Among these, SEM, spike, envelope, and membrane are the part of the outer surface structure of protein. The spike proteins on the envelope help the virus anchor to the host cell. S protein has a trimeric structure with 2 major functionalities, S1 and S2. S1 domain encompasses the receptor binding region, while S2 domain, which is connected to the cell membrane of the virus, which is responsible for cell fission. S proteins are cleaved to S1 and S2 by the activity of certain host proteases like transmembrane protease serine subfamily, followed by virus genetic material passes through the pore of the host cells. So analysis of these things we can find here. Okay, this is all analysis suggested that SARS-CoV-2 recognizes human ACE2 more efficiently than SARS-CoV-1. This is what we found. We are facing the problems today. ACE2 enzymes more efficient than SARS-CoV-1. SARS-1 COVID was there. It has come and it has gone. 2001 it was there. Again, it was identified in 2013. It just went away. But and it's meeting with this ACE2, angiotensin converting enzyme 2, and the SARS-CoV-2 spike protein was shown to have a strong binding affinity to human ACE2. The overexpression of the ACE2 enhanced disease severity in a mouse model of SARS-2 COVID infections, which demonstrate viral entry into cells through receptor binding is a critical step. And that we are facing here. And this ACE2 receptor, all these things are only point is we have to be remembered that TMP, RSS2, P2, and ACE is the main source, or is the main reason for entering the virus into the body, and it goes for a numerical disorders. This is all these things is a common things. It is a common thing. I don't going to read it, but exactly what has vectors. This is there. Just this is yes. 1 minute. Just 1 minute. I am showing this point here. Okay. Yes. Transmission is there. Some people have been infected and recovered without showing symptoms, and such people may be able to spread COVID-19, although uncertainties remain in terms of the asymptomatic transmissions. When the contaminated droplets falls into the floors of the surfaces less commonly, they can remain infectious if people touch contaminated surfaces and their eyes, nose, mouth, and this story we know everything. On surfaces, the amount of the active virus decreases over time until they can no longer cause infections, and surfaces are not thought to be the main way that the virus spreads. It is unknown that what amount of virus on the surface is required to cause infection via this method, but it can be detected for up to the 4 hours of the copper. This also is a known things only. Okay, now let us see this point here. Of coronavirus contains specific genes, that is ORF1, downstream region that encode proteins for viral replication, nucleocapsids and spikes formation. The glycoprotein spikes on the outer surface of the coronavirus are responsible for the attachment and entry of the virus into hot cell. That we have studied. Now, next point is what? It is a receptor binding domain is loosely attached among the virus, and therefore virus may infect multiple hosts. This is, you can see, what is important. Receptor binding domain is loosely attached to the among the virus, and therefore virus can infect in multiple host cells. Other coronavirus mostly recognize aminopeptidases or carbohydrates as the key receptor for entry to human cells, while SARS-CoV MERS recognize exopeptidases. The entry mechanism of coronavirus depends upon cellular proteases, which include human airway trypsin-like protease, cathepins, and transferase protease serine. And this is also going for a peptidase TPP4 virus, in which among these everything I like to say in one word, they are only going for a ACE2, ACE, that is angiotensin converting enzyme 2, is the key receptor of these things. So this is whole cycle is there. Okay? It is going, what chemistry is there? This is all biological processes. Okay? What type of things are going on? This S protein. So our purpose is, as a chemist, our purpose is to binding this one S protein and receptor ACE2. That point we have to study, and we have to prepare a drug molecule based on these compounds, on these mechanisms. That's why I'm telling you. Okay, first of all, we should know the mechanism. Okay, what have the work? What is we are dissecting? I'm telling the word dissecting. Okay, dissecting and understand the mechanisms. On basis of these mechanisms, we can direct or we can find what way the drug molecule can be synthesized. So let us discuss this point. This is also a story. What fission and transcription and vacillations will be, and finally virus release. And this is during this lockdown period, everybody has read this one. Everybody gets aware of these things only. But just I'm telling one compounds, symptoms also I'm not telling this one. But viral testing also I'm not going to discuss it here. But effective chemicals, yes. The earliest effective chemicals, see, people were talking about the chloroquine. Some people are talking chloroquine phosphates. Some people are talking for some the remdesivir. This other numbers of drugs people are finding. But what exactly chemicals are required, that we have to study here. Okay, the earliest reported case of the had a link to a large seafood and animal market in one. This is also, but here this case, the point you can see. This is the things, see people targeting DDPP, chloroquine diphosphates. This point you can write. Health, furthermore, dilution of the health force as a result in the generate management to how to lead increase mortality rate. Okay, this one. Currently, it is delighted to point out that at least 4 kinds of effective chemicals displayed strong antiviral potentials, which have been permitted to combat the disease in a clinic according to the 6th editions of the COVID-19 protocol in China. This is a lopinavir, ritonavir, ribavirin, all this one nucleoside analogs are used for a coronavirus. So in this case, you can see this point. What molecules you are designing, see, if you see the slide, immediately the things comes in our mind. This is a ribavirin, liponavir, ritonavir, erbidol, this chloroquine phosphate, which just start synthesizing molecule. Now it will be effective against coronavirus. Friends, it becomes a story only. We will not get any idea. But actually, what this scientist has done, can direct targeting endosomal acidifications. If you just see the structure here, in this structure, and this target is directing us, you can go for this synthesis. You can go for synthesis. And summation of this one, just do one thing, one drug molecule pick up, orbitol, go for this connection, get a basic nucleus, and this nucleus can be added, can be combined with other molecules, and this synthesis molecule, there's a novel compound you synthesize, and then you find it, what the target molecule, what the effect, what the dose effect I can study. So that is some new directions of the things. Because see, just like I was talking about non-nucleoside, reverse transcriptase inhibitors, every group has shown different concepts. That's why I'm telling you, one CDRI people is showing, one Chinese people is showing something, one medicinal NIH people is showing, one people is showing I am preparing NPTT analog, one people is showing I am horseshoe type structure, one people says I am a heat NHPHU type structure, one people is showing that is of dominant derivatives. It is a trial and error, and that has situation has come in India also. We face the things. One hospital people, they given some chloroquine phosphate become all right. It is not like that. You must be knowing there's a pharmacokinetic and pharmacodynamic study. Some people see becomes all right very immediately, and some people take some time like that only. So right now, it was also earlier, it was a belief lopinavir and ritonavir, doctors used to give these medicines. This belongs to the medication protease inhibitors. So protease inhibitors, and they are giving, and people becomes all right. This was applied for MERS also, Middle East and the retrovirus. This is the drug molecules which is given. China people has given some other drugs. Lots of ribavirin. This is ribavirin is also antiviral prescription medicines approved by FDA. These also given combinations. So like this, all drugs are given, but all drugs has given that has not sufficed the target. Okay, what target it is applied and what target people become sure survive. It is a simple things, and that's why new concept you must be knowing. Concept has vaccination and something has come. That story we don't speak it, but important thing. Just my message is there. Chloroquine is also has also function on different ways. People perception is different. You must be knowing the research paper. Here some people is showing chloroquine analogs. People are doing chloroquine itself only. Some people are going for a phosphorylation. Some people are going for its lipophilicity characters. Some people are going for its pro-drug concepts. Numbers of things people are doing, but no survival is there because the main reason is what? We don't find the active site of the S protein and these enzymes. That is only things. Some people have found that is they are going for simulation study also. Simulation study also, it is also among the features. But I don't say, or I'm not agree that these simulations will suffice or it can be the option of a coronavirus. It is not like that. So these things, I have some other schedule. So I'm going to stop here my representation. These things, please ask me the questions because I can talk both things also. But problem is what? I have some other schedules. I have that I told Dixit Madam also. Please, any question is there? Tell me.
Sir, thank you very much, sir, for such a nice session on anti-HIV chemistry and
Because see, listen, one point. Anti-HIV, if you know, then you can prepare some other medicines, viral medicines. It's a
yes,
basic thing.
yes, sir. It was indeed very interesting and informative, sir. With your permission, we can take few questions.
Please.
Sir, my question is, can we combine this two therapy like any traditional antiviral drug and
Yes.
natural drug like any phytochemical or essential or oil?
Correct.
Synergistic effect,
Yes.
individually or synergistically, is it fine for this therapy?
It can be, because that's why this is the concept of heart only. That's why I told one word, that's heart, highly active antiretroviral therapy. There is no specification for individual drugs. That's why doctors are giving only heart, combination of 2 or 3 drugs. Because you know that virus is there, it gets replicated, new virus is generated. And unless we know the structure of virus, we cannot prepare the medicine. That's why people are going for a mixed therapy. That is correct. Yes.
Then one more question is asked, like what is the reason for failure of remdesivir drug, which was used for COVID-19, this virus, coronavirus, against coronavirus?
Yes. It was there, but it was given. But to somewhat extent, it has some activated, it has stopped the growth, it has some of, it's binded with S proteins, but no more certain results are found. Doctors has advised, doctors has given all the remedies, all the treatments are given, but no survivability is found. And that's why the people has gone for the options that is of a, this one, vaccinations. See, people are going for stimulation study also more.
Okay. Next question I have observed is one person is asking, is there any permanent cure for this viral infection, coronavirus infection?
There is no surety, because what I told in 2002 it has come, again it has come in 2013, and on this earth, number of viruses come and gone. Likewise, if this also happened, coronavirus has come, tomorrow new viruses may also come. So people will forget about coronavirus to COVID-2.
Exactly. Nice. I'll ask, is there any other question from audience? So we have covered few questions. Fine. Thank you very much, sir, for such a nice, interesting and informative session.
You can
Now,
share this PowerPoint, yes.
okay, fine. And now we'll go ahead with the next technical session.
So, okay. Shall I stop, madam?
Madam, is there any next? Yes, sir, please, you can, sir.
Thank you very much. Take care of yourself. Thank you, good luck.
Thank you very much, sir. Just we'll wait for a few minutes for next session. Well, there is one instruction, like we are going to commence next session at 12 o'clock. So participants are requested to join again at 11:45 sharp. 11:45. You can leave at this time. We'll join again at 11:45. Next session is at 12 a.m. Sorry, 12 noon.